Hi everybody, my name is Jessica De La Cruz and today's date is January 16, 2019. We are on week 2 lab that it is called Bacterial, Enumeration, Dilution, and Plate Counts. So in this lab, we are going to observe the exponential growth of microbes through binary fusion. And binary fusion is basically the asexual reproduction when one cell splits into 2 cells. And because microbes have the ability to exhibit exponential growth, just a few amount of cells can multiply into many cells within a short amount of time. So the goal of this lab is for me to get a viable plate count in which I can achieve a CFU, a colony forming unit, of 30 to 300. Okay. So I'm gonna get the pipette from the distilled water. I'm just gonna take everything out. Okay. And then I'm gonna get this and I'm gonna swirl this just to kind of evenly spread out the yeast. Just evenly spread that out. So what I have here is I have my test tube labeled from 10 to 10, 10-1 to 10-6. Oops. Corresponding to their plate from 10-1 to 10-6 as well. Okay. So what I'm going to do is I'm going to grab 10-1 and I'm going to pipette 0.25 of the yeast. Okay, and then I'm going to put that into the first test tube, and I'm going to mix that around, just making sure everything's all in there. Okay, just making sure it's all mixed up in there. Then what I'm going to do is I'm going to grab 0.25 of this. Okay, and then I'm gonna put it into here, the 10-2. Oh. I don't think it grabbed. Okay, sorry guys. Okay, that's 10-2. Put it in there. Oops. And then kind of just mix it up in there. Okay, now I'm just going to get everything out and then get 0.25 of this from the 10-2. Okay. Okay, and then I'm going to put this into the 10-3 test tube. And then kind of just mix that in there. Okay, oh, and I'm going to grab 0.25 of this one. Oh. Oops. Okay, and I'm going to put that into the 10-4. 10-4. And then we're going to mix that in there. Okay. And then we're going to get another 0.25. Okay. And then we're going to put that into the 10-5. Okay. Let's mix it all up in there. Okay. And we're going to get lastly 0.25. Okay, and then we're going to put that into the last one. Just get it all mixed up. Okay. Then now what I'm going to do is I'm going to just rinse this out. Make sure everything's out. I'm rinsing it in the distilled water. So now what I'm going to do is I'm going to get each of the test tubes, and I'm going to put 4 drops of each of the solution into the corresponding agar plate. So I'm going to get this. I'm going to get this. And now the experiment says to put 4 drops. So what I'm going to do is I'm going to turn over the plate. I'm not going to open it I'm not going to open it too much just because I don't want bacteria to get into there. So I'm going to do 4 drops. 1, 2, 3, 4. And then I'm just going to clean that out. And then I'm going to swish it around, making sure that it gets all over the agar. Okay. All right. So I'm going to do this for all 6 of the plates. And I'm going to leave it like this I'm going to place, once I'm finished, I'm going to place this into my incubation location for 12 hours. After 12 hours, I'm going to invert the plate and let it sit for an additional 36 hours. And then it'll be on the next video where I will show you the results. Bye guys!